Hi everyone, this is Sharon. In this video, I'm going to talk about the field of pharmacogenetics through the case study of anticoagulant medicine, Warfarin. Pharmacogenetics might seem like a hard vocabulary, but if we separate this word into three parts, pharma, co and genetics, then it will be quite self-explanatory. Pharma means the study of medicine, co means together, and genetic means the study of genes. So altogether, pharmacogenetics means the study of how drugs and medicine interact with our specific genes. I'm going to investigate into an anticoagulant, which is a drug used to prevent blood clots, specifically Warfarin. I'm especially interested in this drug because I once read an article about a pharmaceutical company being sued for selling Warfarin, which leads to pregnant women bleeding or even giving birth to deformed babies. And later, after I did more research about this drug, I then discovered that Warfarin is quite popular. Over 30,000 Taiwanese take Warfarin. Even more, I found out that Asians, including Taiwanese, have a higher risk when taking Warfarin. This is why I chose this specific topic to research on. In my following video, I will be covering the mechanisms, drug safety issues, adverse drug reactions, tailoring treatments, identification of optimal dosage, SNPs, and disadvantages of Warfarin. I will also tell you why more and more people are studying pharmacogenetics. Okay, so now I'm going to do some brief introduction about this drug Warfarin. Warfarin is first used as a rodent killer drug because it can make mouse bleed to death. The mechanism of Warfarin used today is shown in this figure. Warfarin blocks vitamin K oxide reductase. Notice the ASE. Vitamin K oxide reductase is an enzyme that creates vitamin K. Vitamin K plays a vital role in blood clotting, which locates in the liver, because it acts as a cofactor for the blood clotting factors 2, 7, 9, and 10. So Warfarin simply blocks the production of vitamin K, further disrupting the clotting process. First of all, Warfarin has two enantiomers, the R weak form and the S active form. Enantiomers are two molecules that are mirror images to each other. Think of our hands as an example. They are mirror images to each other, and when we overlap them, they won't fit perfectly. The R Warfarin spotted line, shown in the orange circle, is like my left thumb sticking out, and the S Warfarin dotted lines, shown in the blue circle, is like my right thumb sticking backwards. So our hands are basically a simple analogy, a simple model for the Warfarin enantiomers. The enantiomers also play a significant role in Warfarin. The enantiomers are a main cause for a lot of problems in Warfarin, including drug safety and adverse drug reactions. I will start with how to improve drug safety first. Pharmacists need to effectively purify the enantiomers of Warfarin. We want the S form instead of the R form because the S form has a higher clearance rate in our body and is 5 times more potent as an anticoagulant. By producing the Warfarin, both enantiomers will be produced due to their nearly identical structure. So pharmacists need to purify the drug by picking out the R form. This ensures the drug safety. Because the R Warfarin's lower clearance rate, adverse drug reactions of bleeding, cell necrosis, osteoporosis, purple toe syndrome, and calcifications might occur due to the prolonged period of the R Warfarin staying in our body. It takes 37 to 89 hours to get rid of R Warfarin in our body through urine, and that's quite a long time. Another problem associated with Warfarin is that it is hard to control the dosage for each person because each individual's genetics vary, and this seemingly slight variation actually plays a huge role in deciding the Warfarin dosage. Overdosing leads to strokes, while underdosing leads to thrombosis. So pharmacists need to tailor treatments to meet patients' distinctive genetics. Pharmacists look at factors including genes, height, age, weight, and health experiences to help them decide the unique dosage for their patients. For example, I found a study conducted by the Academia Sinica in Taiwan, trying to measure the optimal dosage of Warfarin for each individual via their genes. Ethnicity, like in the alcohol case we studied during class, also plays a very important role in deciding the optimal dosage for Warfarin. As I mentioned before, Asians tend to have a higher risk when taking Warfarin. This is taken into optimal dosage consideration as Asians need lowest dosage, while African Americans need the highest, which are shown in this graph. Another thing we can learn from this graph is the diversity of each ethnic group. Since Caucasians have the narrowest interquartile range, shown in the blue bracket, and Hispanics have the widest interquartile range, shown in the orange bracket. This graph shows that Caucasians are the least diverse and Hispanics are the most diverse in terms of their genetics. Pharmacists use a variety of dosage tools to help them decide the optimal dosage for each unique person, which shows how important the tailored treatments and identifying the optimal dosage are for the medicine warfarin. This case study of warfarin also associates with SNPs, which we have discussed in class. SNPs are single nucleotide differences in the members of the same genome, which leads to the formation of different alleles. CYP2C9 and VKORC1 are proteins that play a significant role in warfarin's mechanism, and there are 3 SNPs, 2 located on CYP2C9, associating with warfarin metabolism, and 1 located on VKORC1, associating with VKORC1 production. For CYP2C9, there are 3 different alleles. The first has the highest rate of metabolism, while the second and third has only 70% and 30% of metabolism rate, respectively. The lower the metabolism rate for S-warfarin, the lower dose is required. Agents tend to have the protein with a lower metabolism rate, so that's why we need to take a lower dose than other members of other ethnic groups. There is another SNP on the VKORC1 protein, which the gene with the A base produces less VKORC1, while the G base produces more. VKORC1 provides instructions to create vitamin K epoxide reductase enzyme, so more VKORC1 means a requirement of a higher dose. The A base, more prevalent in Asian communities, respond to a lower initial dose, which also explains why Asians need to take a lower dose. To sum up, this graph shows the interaction between the dosage amounts with the different alleles of these 2 genes. VKORC1G means more dosage, and CYP2C91 means more dosage as well. However, there still are some disadvantages for pharmacogenetics. To start with, it is money consuming. Pharmacists have to use a lot of different tools to help them modify the drugs. All those different drugs cost a lot of money as well. Also, it is time consuming. Since every individual has different conditions, pharmacists have to take time to tailor treatments for each person, so this costs a lot of time. So after talking about the case study of warfarin and its association with pharmacogenetics, I guess it's time to talk about why more and more people are interested in studying pharmacogenetics. This case study of warfarin is just one example of the applications of pharmacogenetics. Nowadays, more and more genetic engineers are devoting themselves into this field of science because we now have more knowledge about our genetic information. More and more researchers are finding ways to cure diseases by creating new drugs or improve the already existing drugs. So this is why pharmacogenetics has becoming more and more popular. Pharmacogenetics is no doubt an excellent field of science with much potential, which can be shown through my case study of the anticoagulant warfarin. Even though there are a couple disadvantages to this field, like limitation of money and time, more accepting this new genetic engineering technique as it can help solve problems associated with the interaction between drugs and our genes. I believe this field of science has endless potential in the future and can continue to benefit humans. So that's all. Hope you all now understand the mechanisms of warfarin and its applications in the field of pharmacogenetics. Thank you for watching! The aforementioned research has come from reliable sources such as master and PhD journal articles, and an Academia Sinica study, as shown in the works cited list. Thank you again!